Let us now proceed to the next part of the chapter, that is cell division. Cell division is one of the very important characteristics of the cell and living organisms. Cell undergoes division to form daughter cells is called as cell division. Due to cell division, a multicellular organism is formed, growth takes place, wound is healed, and emaciated body can be restored. A new individual is formed from the pre-existing one only due to the cell division by the process of reproduction in which cell division plays a vital role. There are two types of cell division. The first one is mitosis, second type is meiosis. Mitosis occurs in the somatic cells and the stem cell, while meiosis occurs only in the germ cell. We have already studied the structure of chromosome in the previous class. Let us recall the structure once again. Each chromosome is made up of DNA and it appears dumbbell shaped midway during the cell division. There is a constricted region on each chromosome. It is called as the primary constriction or centromere. This divides the chromosome into two parts. Each part is called an arm. The centromere has a specific position in each chromosome. We have studied the structural organization of the cell. We know that each cell has nucleus and many cell organelles present in the cytoplasm. Before any type of cell division, the cell prepares itself and in this process doubles up its chromosome number present in its nucleus. That is, if the number of chromosome is 2n, then it is doubled to 4n. This phase is called as the interphase of the cell division. Mitosis. Somatic cells and stem cells divide by mitosis. The mother cell which undergoes cell division is 2n diploid, which means chromosomes present in nucleus are in pairs. Before cell division, chromosomes are duplicated and the number are doubled up to 4n. The whole process of mitosis is completed in two main steps. The first step is karyokinesis, that is division of nucleus, and the second step is cytokinesis, that is the cytoplasmic division. Division of nucleus, that is karyokinesis, is completed in 4 steps, that is prophase, metaphase, anaphase and telophase. Let us learn about them one by one. Prophase. Condensation of chromosomes with sister chromatids takes place. Centrioles duplicates and move to the opposite poles. Nuclear membrane and nucleolus starts to disappear. Metaphase. Nuclear membrane completely disappears in metaphase. Chromosomes are arranged in the equatorial plane of the cell. Spindle fibers and astral rays are formed by the centrioles. The spindle fibers get attached to the centromere of the chromosomes called as spindle apparatus. Anaphase. The centromere splits as the spindle fibers undergo contraction. As a result, the cystochromatids separates and are pulled apart to the opposite poles. The separated sister chromatids are the daughter chromosomes. Telophase. The daughter chromosome starts to decondense to form chromatid reticulum. Nuclear membrane and nucleolus are formed around the newly formed chromatin reticulum. Spindle fibers completely disappears. Cytokinesis. In plant cell, cytokinesis takes place by the formation of cell plate from the center of the cell to the periphery, dividing the cell into two daughter cells. In animal cell, a central notch is formed that deepens towards the center and divides the cell into two daughter cells. Let us study the second type of cell division, that is meiosis. Meiosis takes place in the germinal or reproductive cells. One cell divides to form four daughter cells called as gametes. Male gametes, that is sperm, and female gamete, that is ovum. It is called as reduction division, as the chromosome number is reduced from diploid 2n to haploid n. It includes two stages, meiosis I and meiosis II. Meiosis I. It is a reduction division. The chromosome number reduces from diploid 2n to haploid n. It includes two stages. Karyokinesis 1, that is the division of the nucleus, and cytokinesis 1, that is the division of cytoplasm. At the end of meiosis I, two haploid daughter cells are obtained from one diploid cell. Karyokinesis 1. The division of nucleus takes place in this stage. It includes four phases, that is Prophase I, Metaphase I, Anaphase I, Telophase I. We'll study all the phases in detail. Prophase I. This phase includes 5 stages and it is the longest phase. The 5 stages of Prophase I are leptotene, zygotene, pechete, diplotene, and dikinesis. In the leptotene stage, chromosomes are duplicated and found as thin thread-like structures. In zygotene, pairing of homologous chromosome called as synapses takes place. In pechete stage, the non-sister chromatids of the homologous chromosome attach at certain points called as chiasma. At chiasma, genetic material DNA is exchanged between the non-sister chromatids and this process is called as crossing over. In the diplotene stage, crossing over is completed and homologous chromosomes separate from each other except at the chiasma. Metaphase I. Chromosomes align together in the equatorial plane of the cell. Spindle apparatus is formed. Anaphase I. The two homologous chromosomes get separated and move towards the opposite poles of the cell due to the contraction of spindle fibers. As a result, reduction in the number of chromosomes occur. Telophase I. There are haploid number of chromosomes on both the poles of the cell. The nuclear membrane and nucleolus are formed. Cytokinesis I. In plant cell, cytokinesis takes place by the formation of cell plate from the center of the cell to the periphery, dividing the cell into two daughter cells. In animal cell, a central notch is formed that deepens towards the center and divides the cell into two daughter cells. Meiosis II is just like mitosis. In this stage, the two haploid daughter cells formed in meiosis I undergo division by separation of recombined sister chromatids and four haploid daughter cells are formed. Process of gamete production and spore formation occurs by meiosis. In this type of cell division, four haploid, that is N daughter cells are formed from one diploid, that is 2N cell. During this cell division, crossing over occurs between the homologous chromosomes and thereby genetic recombinations occur. Due to this, all the four daughter cells are genetically different from the parent cell and from each other too. With this, we have studied both the types of cell division.